So considering the materials we have left for the course, we need to leave the gene regulation and microRNAs for the introduction to genomics topic, and actually that's where it should be because we have the regulatory data of Ensemble, and it's more of a genomics topic. So what we have for now is protein sequence analysis, and for next week we'll have the topic of gene ontology and the gene enrichment analysis with a very simple test data set. But anyway, we need some text is misplaced, but let's start. So we'll talk about what kinds of tasks we have for single sequence analysis. As the title slide tells you, this is web services for single sequences, which you might then upscale if you download the corresponding programs into your own computer, or if you write a script which is able to use the programmatic interface of the services. This is available at least for the subcellular localization prediction, so that you could send thousands of sequences one at a time or a few at a time. But they also accept small batches of sequences up to some dozens of sequences and some thousands of sequence characters, so we can easily do a batch of 15 sequences, which is in the homework. But we'll look at the topics of sequence analysis program types, and then we'll discuss sequence motifs and have a look at sequence motif databases, which is a very useful tool for identifying functions of proteins. Okay, so many analysis programs are strictly for a single program. Some accept multiple sequences as their input. Some require multiple sequences, like multiple sequence alignment. And then you have to really think so that you don't make a stupid mistake of sending a DNA sequence to a protein sequence alignment, to a protein sequence analysis program, and or vice versa. Okay, some programs give you statistics of your sequences, and some take your sequence and then analyze it against something inside the analysis service, such as a motif database. And it's important that you really have your eyes open when you do the analysis. It's so easy to just drop a file into an analysis tool, but you really, if you want to have serious results, you really need to go into the documentation and see that you are following all good practices, that your data is as expected for the program, and you need to look carefully into the results so that there are no funny things there. For instance, we had a case where somebody inserted a FASTA formatted file in a molecular weight calculator which was accepting raw sequence only. And luckily the program was reporting the sequence as entered by the user, and when the first letters of the sequence were HUMANCA, so human CA, so you could see that, oh, the sequence description was there, and the sequence was actually longer than it was meant to be. And of course, the molecular weight was off by being too large. Okay, then in addition to having right kind of input and making sure that you vet your articles, or you have an authority like your bioinformatics teacher who tells you that this is a good and reliable tool that has been around for a long time. So you need to see if your input is sufficient. So for instance, if you have a promoter analysis tool, it's not enough you give the coding sequence because the promoter is in front of the coding sequence. In some cases, the program says explicitly that the sequence must be at least 50 amino acids long if it's trying to find a feature which is in the first 20 or 25 amino acids. So just making sure that you are not going blindly into the results. And then if you are looking at the results and it's a table that has all kinds of columns that have numbers you don't exactly understand, it's a good idea to read the documentation and explanation and see what is the actual result. You want to see what is a control value. For instance, the subcellular localization program for signal peptides gives a reliability value from 1 to 5. And I have to check every time if 1 was the best or 5 was the best. So these things are things you just can't know. You have to make sure you read it from the documentation. For many popular kinds of program like prediction of transmembrane helices, there are benchmarking articles which are making a critical comparison of different programs that are available, and they evaluate the results of their work. And again, sometimes you need to be critical if for instance the benchmarking article is using a set of sequences which was used in training of one of the programs. So obviously it will be very successful with the training examples. But anyway, reading is the key, as for a lot of science here. Okay, for DNA analysis, you may have been using these things to help your lab work, like finding restriction enzyme cut sites to make calculations of expected restriction length fragments. Or if you need to make primers to make the polymerase chain reaction and amplify your DNA, so the primer design programs try to find primers which will bind in the lower temperature and be released in the higher temperature of the normal PCR temperature program, and the reverse and forward primers need to be matching in the melting temperature or annealing temperature of the sequence, so that you get a maximally efficient amplification. And obviously the primers can't be complementary, so that they would be just binding to each other and not your target DNA. Okay, the analysis of transcription factor binding sites will come up in our genomics course, and actually I should remove it from the theoretical DNA analysis because it's much more efficient if you do an experiment and you have experimental transcription factor binding sites or DNA regulatory sites. But there are tools for predicting those sites as well. For proteins, you can do quite a few things like virtual digestion. You can predict the pattern of peptides you would get for a mass spectrometry analysis, which is usually performed on a trypsin digested protein. And there are all kinds of properties you can calculate along the sequence. You probably have heard of the running average calculation in the numbers of coronavirus infections. So instead of giving very highly variable daily numbers, you could give numbers for the last 7 days and have the running average over time. In the same way, you can do running averages over a number of amino acids in the sequence, and in that way see how a property is changing over the sequence. There are many structural prediction programs. We will not discuss the prediction of secondary structures or complete three-dimensional structures here. That would be for structural bioinformatics, but we do take a look at transmembrane helix prediction tools. And then there are many signals which are easily detectable by bioinformatics tools because they have a large concentration of a specific type of residues in a short region of the sequence. For instance, the signal peptides, which have a stretch of hydrophobic residues followed by a break into some charged residues, and these are pretty well predicted from eukaryotic sequences. And there are other localization signals, mitochondrial targeting peptides, etc. Okay, so as I indicated, the running average or calculation window approach is usually used for the sequence property profiles, and hydrophobicity is something that is commonly used, and that's an easy way of getting a quick look at potential transmembrane helices if you have a single protein of your own interest which you want to see on your own, so you can get those things. You can calculate charges on a window calculation and so on. But anyway, we'll look at an example of hydrophobicity profiles, and this would be your first approach in finding signal peptides and glycosyl phosphoinositol anchors, which are also of hydrophobic character, but they are in the end, in the C terminus of the sequence. And the transmembrane helices can be anywhere in the middle of the sequences, and we know that you need an alpha helix of approximately 20 amino acids to create an alpha helix that goes through the membrane bilayer. And the normal rule for detecting a feature is to select a window which is about half of the expected window size. So 9 or 11 could be good for this analysis. And now here's the analysis of hydrophobicity of a rat olfactory receptor. And I'm not sure if you see it from the screen, but at least you will see it if you open the PDF and go to slide 9. This is made with a tool called ProtScale, which is able to use dozens of different parameter sets for the property you want to calculate, and it has all the tools for setting the window size, etc. And now this is hydrophobicity, meaning that hydrophobic is up in the high values and hydrophilic is down. And it would seem that there are indeed quite a few peaks here, and if we assume that maybe 0.5 is a reasonable threshold for finding transmembrane helices, so we would have 1, 2, 3, 4, 5, maybe 6, 7, 8 peaks here. And you may have heard of 7 transmembrane helix bundles of sensory receptors, G protein coupled receptors. They always have the same structure of 7 transmembrane helices, and we looked at the database of 7TM receptors. This is one of them, but we can't really figure out where the 7 helices are. So let's try to see if we can run a demo at the server, which is in the Swiss Institute of Bioinformatics. If you want to try it with your own laptop, feel free to go to the same site and copy the ID from UniProt, and let's see how this goes. I'll change into screen sharing. There we are. And copy the link from here. And I'll copy the SwissProt UniProt accession code. And we want to do hydrophobicity, and there are really many different scales which are not that different. You can see that there's more than 10 scales for hydrophobicity, but Kite-Doolittle is one of the classical ones. And if you want to see what the values are, so I clicked to open the link in a new tab, and there we see it. So the very strongly positively charged arginine has the lowest value, -4.5, and then some of the hydrophobics, valine, isoleucine, leucine, have values that are on both sides of +4. Phenylalanine 2.8, tryptophan, which is actually not that hydrophobic in this scale. But anyway, let's see. Let's see how the calculation goes with window size. Let's do it with window size 3 first. And now, spoiler alert, we get the transmembrane helices picked up from the UniProt headers so that we see where they should be. And now, I hope you can see, let me increase the size a little bit. I think you can see that the result looks less clear than the example I had in the slides, which was done with window size 9. Now with window size 3, there is too much variation. You don't see the trend of hydrophobicity so strongly. We might have a few kind of strong continuous regions of hydrophobicity, but not as clear as it could be. So let's try with 11. And now this starts to look quite a bit clearer here. 3, 4, 5, 6, 7. So if we ignore these lower peaks and this very narrow peak, we might have our 7 helices. And let's look at the helices as documented previously. We might tend to guess that these are not real peaks, but the highest ones are, so that we wouldn't have anything which is falling between 60 and 100 something. Let's see if this confirms. Actually, no. So the predictions with a better program have 39 to 60, and then 80 to 100, 140 to 177. Oh, sorry, 120 to 139. We should be looking at the helical segments and the transmembrane features. We also have the topological domains, which are predicted to be cytoplasmic and extracellular as the peptide chain is snaking its way from in to out, out to in, etc. Okay, so this is a way to do these protein scales. We could try to do other things here as well. We could do a prediction of average buried area, but again, that is very similar to hydrophobicity and doesn't really work for membrane proteins as well as for soluble, water-soluble proteins. So let's go on with the slides here. So would it be more efficient if we do a real transmembrane helix prediction?